All right. Hello, everyone. Welcome back. Again, we are here at the 2022 SLU Summit for Water Resources in a Changing Climate. Thanks so much for joining us today. I'm very excited to introduce our next presenter, Binbin Wang. Dr. Wang is one of our postdoctoral research associates here. He is doing work with the Water Institute, as well as the Biology Department in the College of Arts and Sciences here at St. Louis University, as well as with NGREC, the National Great Rivers Research and Education Center. So he is going to present today on ecological effect of thiamine on the primary producers in aquatic environments. And with that, I will go ahead and hand it over to Dr. Wang. Thanks so much for being here.
Yeah, thank you, Rachel. Hello, everyone. I'm Binbin Wang. I'm a postdoc from Water Institute. Thank you, Rachel, for the introduction. And the title for my today's presentation is the ecological effect of thiamine on the primary producers in the aquatic environments. So first, I want to have a brief introduction to the primary producers that my research is focusing on in the aquatic environments. So I'm focusing on the algae, like phytoplankton and periphyton. So algae produce almost half of the oxygen on the Earth, and they are the basic part of aquatic food webs. And they also provide food for small and large animals. Algae also recycle nutrients and absorb CO2 from the air. So the growth of the algae is very important to mitigate the influence of the climate change. But algae also cause problems like harmful algal blooms. I bet you may have heard that for many, many times. So another one I want to introduce is thiamine. So thiamine is also known as vitamin B1. It is an essential nutrient to all living things. And the left graph is the molecular of thiamine. And thiamine works as a cofactor in life-sustaining enzymes that are required by basic cellular metabolisms. So the right figure shows three very basic metabolic processes occurred in the living cells. So they are citric acid cycle, pentose phosphate pathway, and glycolysis. So I'm not going to the details of each step of these processes, but just show the significance of thiamine to all the living things. So how does thiamine and algae connect to each other in my research? So that is because thiamine is required by all the living cells, but not all the algae, not all the primary producers in the aquatic environment can synthesize thiamine by themselves. So here's a summary of a research. It found that almost 30% of the algae surveyed in this research cannot synthesize thiamine. So that means they have to get thiamine from the environment. So the next question is there enough thiamine for the periphyton to rely on? So the answer might be not, at least in the marine environment. So there is depletion of thiamine in large areas, in the coast areas, and also in the open waters in the ocean. But there are plenty of phytoplankton rely on thiamine. So there seems to be thiamine limitation in the oceans. And notably, most harmful algal bloom species, so they need thiamine. So this gives me some thought, like, so maybe we can control or mitigate the occurrence of harmful algal blooms by controlling the concentrations of thiamine in the environment. And the algal blooms happened in the fresh waters. So what is the situation of thiamine in the fresh waters? Unfortunately, there's just a couple of research mentioned the thiamine in the fresh waters. And they found that river discharges can influence thiamine's concentrations in the coastal waters. But for the status of thiamine in the fresh waters, we still know very little about that. So because of that, I want to investigate the ecological roles of thiamine in the fresh waters. And I want to answer two questions. And like to start the research of thiamine with two questions. The first is there thiamine limitation of primary producers in the fresh waters, just as that happens in the marine systems? And if there is thiamine limitation, so how does the limitation varies spatially or temporally? So to answer these two questions, I designed an experiment. So I conducted nutrient enrichment experiment with the nutrient diffusing substrate method. I made the substrate and also spiked nutrients into the substrate and put the substrate in the streams and leave that in the streams for 13 times, and then collected the nutrients will diffuse from the substrate to the stream waters, and the primary producers, mainly algae of periphyton, can attach to the substrate and use the nutrients diffusing from substrate to grow. So after the incubation, I will sample the biomass of the periphytons growing on the different treatments, and to compare the biomass. And by doing so, we will see the effect of the nutrients spiked in the substrates. So the biomass I measured is chlorophyll A. Just use that as a proxy of the biomass of the periphyton on the substrates. So here is the results, one of the results. So in this figure, the x-axis, the concentrations of nutrient spiked in the substrate, and the y-axis is the biomass, that is the chlorophyll A. So the addition of thiamine from the first figure, the addition of thiamine can promote the growth of periphyton on the substrate. That means there is not enough thiamine in the stream naturally. So that also means there is thiamine limitation in the stream. So this is the logic. So in the second graph, we can say, okay, nitrogen also promoted the growth of periphyton on the substrate that with addition of nitrogen. So there is also nitrogen limitation in this stream. And phosphorus did not have any promoting effect to the growth of periphyton. So that means there is no phosphorus limitation. So, but this experiment just measured the effect of thiamine or nitrogen at a one-time point. So in order to make sure this is not a random effect, just happened once, I conducted another experiment with a longer incubation time. So I elongated the incubation time to 32 days. So just as the x-axis shows in this graph, I sampled the biomass at different time intervals. I sampled on the 2nd, the 4th, 8th, 16th, 24th, and the 32nd days of the incubation, and to see the change of the biomass during the incubation, and to make sure that the limitation did not happen just once, just randomly once. So it is very clear here. The black line is the biomass of a periphyton on the control, and the purple line is on the nitrogen addition treatment, and the blue line on the phosphorus addition treatment, and the orange line on the thiamine addition treatment. So it's clear that from the 8th day, there is, we can observe at least thiamine limitation and also nitrogen limitation. And after we observe it, the limitation lasted until the end of the experiment. So now we are confident to see that there is thiamine limitation in this stream, in the fresh water. And here comes another one. So does thiamine limitation vary temporally or spatially? Before answering this question, I want to introduce another term. So it's the effect size. It equals the biomass on the treatment, nutrient addition treatment over the control and -1. This is a standardized term that can be used to compare the results of the nutrient diffusion substrate experiment that are conducted in different streams and also conducted at different times. So here is the result of this experiment. I tested the seasonal variation in 2 streams, and the left one is in the nameless brook. So in this graph, the x-axis are the months when I conducted the experiment, and the y-axis is the effect size. The symbols, the triangles, that is the effect size of nitrogen, and the black circles are phosphorus, and the crosses are thiamine. And the stars above the symbols represent there is a significant effect from the addition of this nutrient. That also means there is a limitation by this nutrient. So from June through October, there is thiamine limitation, but the effect size varies a little. And there, to contrast, there's just a nitrogen limitation in June and August, but not in October. And there's no phosphorus limitation in nameless brook. And in the second stream, the Pico Creek, so the graph on the right, there's also thiamine limitation in August and October, but no nitrogen limitation. So this stream, is not a nitrogen limiting stream, but phosphorus limitation happened once in October. It might be a phosphorus limiting stream. So I also tested the interannual variation of the limitation. And I conducted the same experiment, the experiment at the same months, but in different years, from 2015 through 2017. And there is a thiamine limitation in 2015 and 2017, but not in 2016. So it seems there also interannual variation. And the nitrogen limitation also occurred in 3 years, but its effect size varies, and no phosphorus limitation observed. So we know that there is temporal variation, but how about the spatial variation? Yes, there is also spatial variation. So I conducted more experiments in more streams. In all the 4 streams, thiamine limitation was observed, but nitrogen limitation only occurred in the nameless brook. So it seems thiamine limitation is more widespread than nitrogen limitation, and the phosphorus limitation did not happen in any of the stream. So it seems when we pay attention to the nitrogen limitation or phosphorus limitation, when we studied or considering algal blooms, controlling algal blooms, there might be some other factors we have neglected or we just lost them. So this is the findings that I want to confirm. So there is thiamine limitation in the streams, and it varies seasonally and annually. So as nitrogen or phosphorus, and the limitation is kind of similar as that people found in the MER systems. And the promoting effect of thiamine addition could be comparable to that of nitrogen. And the thiamine limitation is widespread in the streams, and it occurred more frequently than nitrogen limitation in streams when nitrogen is also limiting. So that's just the start of my experiment, and I want to figure out more about the ecological rules of thiamine in the aquatic ecosystems. Here are some thoughts. So first, how do the physical chemical factors influence the ecological rules of thiamine and its precursors? So because thiamine can be synthesized by the organisms, it can also be degraded by like the UV light or higher temperature. So how So that will be related or changed when the climate change or seasonal changes. So how will that influence the ecological rules of thiamine? That will be very interesting, but we still don't know that in the freshwaters. And how does thiamine and its precursors influence the microbial community compositions? So as I mentioned above, some algae can produce thiamine, but a lot of them cannot. So when the concentrations of thiamine changed in the environment, so the relationships, whether they are competition or the competition of these organisms might be also influenced by thiamine. And how does thiamine limitation of phytoplankton producers or primary producers influence the aquatic ecosystems through food webs? Because thiamine deficiency was also observed in fish, in birds, and fish and birds also die from that. So is there some correlation between the thiamine limitation to the primary producers and the deaths of these animals? That is also still a question we have to answer. So, and also the fourth one, how does anthropogenic activities influence the ecological rules of thiamine? So do we release thiamine to the rivers just as what we did like maybe 20 years ago to the nitrogen or phosphorus, when we did not realize they are also pollutants to the environment? So when we didn't know that, we just released those nutrients and a lot of algal bloom occurred. But how about thiamine? Maybe it's also a factor that we have to pay attention to. So that's all what I want to share with you today. Thank you. And I can take any questions.
Thanks so much, Dr. Wang. This was really interesting. Appreciate you sharing your research. We will hang on for just a minute or two and see if any questions come in. If you have questions for Dr. Wang regarding this research, please go ahead and type them into the chat or the Q&A. It looks like we have one coming in here for you, Binbin.
Yeah, I saw a question from the chatting board. Yeah, so for freshwater sources, did you only look at streams or lakes too? So for this experiment, I only look at streams, but the lakes, I also want to add that to my future research.
Any other questions for Dr. Wang?
Oh, here's another one. Which stream did I conduct the study on? So I conducted this experiment in 4 streams in the Adirondack areas in New York State. So all the results are from those streams. Okay, so which for my future studies, which questions did I want to approach first? The one I'm studying on is the influence of thiamine to the community composition of the algae in the streams. So I was focused on that one. And also I wanted to measure the concentrations of thiamine and the distribution of it in the streams and the lakes. Yeah. Okay, will water temperature changes impact thiamine content in the streams? The quick answer is we are still not very clear to this, but the temperature has potential to change that because thiamine can be degraded when temperature increases. And also the increase of temperature may influence the activities of some algae that can synthesize thiamine and release them to the waters. So that may change the concentration of thiamine in the water. So is thiamine released during decomposition or excreted? So it released during excreted.
Great. Okay. Thank you for all of the questions. We'll give it one more minute and see if anyone else has any other questions here for Dr. Wang. Okay, that is going to conclude this research session. Thank you so much, Dr. Wang. It was great listening to your talk. And thanks everyone for attending and participating. If you have any further questions for Binbin, you can type them in the chat and he may be able to answer them later today. Or you can also always email us at water@slu.edu and we can work to get you connected with Binbin that way as well. If you want to stay in the conference, we will be leaving the social lounge open until 2:15, and at 2:15 we will broadcast our next session, which will be the panel from Research to Impact, Academia in Our Changing Climate. So please stay tuned for that, and we will see you all shortly. Thanks again, Binbin.
Yeah, thank you, Richard.